we're talking about membranes and the lipids that make them up. And for membranes, for a function, some of the structure about membranes is we have to talk about the proteins that are embedded in them, which actually cause function. Each membrane that we're talking about has a specific collection of proteins that create particular functions. All right. If you take a look at it, some proteins have almost no protein, and some membranes have lots of protein. Myelin, which is really a protein that surrounds a nerve signal, which is really controlling about whether ions move across it, have almost no proteins. The only proteins really they have are the ion channels that control that, because we are only moving ions across. Most cells have about 50% protein, 50% fat, or the lipids themselves, and upwards of the membranes that we talk about in the mitochondria, you can have as much as 75% protein in there, because remember, all the processes happen inside the membrane-bound proteins. So depending upon what you're looking at, there's a diversity. Two different classes of proteins and how they're attached. One is integral and one is peripheral. Integral is that it is embedded into the membrane itself. A lot of integral proteins will be both on the surface of the outside and inside of the cell, and they usually span that bilayer. The peripheral ones are attached to the surface, a lot of times through electrostatic interactions with other membrane-bound proteins, or they can be attached very gently with maybe one or two lipids that are embedded into that membrane itself. Here is kind of an experimental condition where the book tells you and then what we would really like you to be able to do later. Okay? Here the book has arrows, and you see all the arrows go this direction. And so you see a mental picture and you see what's happening, and then you see the data. In real life, when you are actually going to be studying past this, we give you that. And we hope that you are able to create the picture backwards by what the data says. So this particular experiment has really very little to do with what I'll have you do on an exam, but I want you to think through this process, okay? We have two reagents that are going to interact with a membrane, and it's going to react with the proteins within the membrane. There are three separate proteins. Now I can see that data because on the SDS gel I separate 1, 2, 3. There's one absent here, but I have three proteins that are different sizes. Now you can see that in these I do a treatment with one and a treatment with other. This treatment with the AE is a reagent that will react with proteins, but it's able to translate through the membrane. The IAE is able to react on the outside but cannot go through the membrane. Okay? And so we can see that if a reagent cannot go through the membrane, it will react with two proteins. If a reagent can go through the protein, it reacts with three proteins. But if I just look at this, okay, I see that, this is darker than that, right? But is it really darker, okay? Internally, this band and that band are similar, okay? So the amount of labeling, the reagent reacting to protein, is pretty much similar between the two, so they're kind of one equivalent. If I look at these two proteins, it looks like they're pretty equivalent, right? But what about this one? That looks what? Much darker, doesn't it? Okay. It looks like in relationship to these two, this one got labeled more. And if I'm looking at relationship of these two, they got reacted about similarly. Okay. How would I interpret this data into a picture of what might be happening? Taking the two cells that I split apart into two different test tubes, they're the exact same cells. One treatment with the reagent that cannot go through would react with every protein that is on the outside of the cell, has protein on the outside of the cell. And they get labeled at about the same rate because they are exposed equally. I do not see the third one because it's internal. Okay, I don't see it at all. But with the one that can go through the membrane, I react both on the outside and I can react on the inside, and you can see that one protein is on the outside but doesn't reach the inside. One protein's on the inside, doesn't react on the outside. That's why I have a difference here. And one goes on both sides, which shows a doubling of its reactivity and the gel. Now, we hope that at some moment in time that you're able to go this direction, and from data given to you this way, you can make a mental picture of what that means. Are you there yet? No, you get that journal article and you read the first line and don't understand 5 words, right? You've been in that position, okay? You don't give up. What do? Google, right? Okay, at least you have a strategy by which to solve this, all right? But as you become more familiar with more and more concepts, we want you to get familiar, or we want you to get to that place. In actuality, in molecular biology or Biochem 3, the textbook that Dr. West uses is all of those things that you learned in genetics in terms of how DNA is processed, but we go back to the original papers and the original data set to show you why, and the experimental evidence, why we've made those models. And so we're going to build you up to that point, okay? Now, this particular experiment, nothing in terms of, I'm not going to ask you a question about it. I'm just showing you where I want you to be at some point in time. Now, remember when we study proteins, we always want to isolate that one protein and study it. The problem is proteins that are embedded in membranes love hydrophobic solutions. They want to be in hydrophobic. The problem is all of our techniques to isolate have been in water or aqueous solutions. They do not like to be in aqueous solutions, so what we have to do is extract them in a way that we get them to make them soluble but don't destroy them, because a lot of times when we add detergents like SDS, we're unraveling them completely and doing analysis and they're dead. And so what we do is we do use a little bit of detergents. The reason why, these proteins that are embedded in the membrane, when I give them a detergent, remember a detergent is made this way, one long hydrophobic chain and one very polar area. And I will lay the hydrophobic chain, okay, along the hydrophobic nature of the protein. Now this protein had a soluble part at the top and a soluble part at the bottom, but this middle had to be coated with all of those hydrophobic chains, and then I substitute these polar heads. So now polar, 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 soluble, whereas this was polar, polar, hydrophobic, not soluble in water. Now we probably will not use SDS, but I use that because you know what that is. You can buy different detergents that aren't as strong as SDS, more mild detergents to try to extract. To actually develop a protocol to extract your protein out of a membrane is kind of magic, or research. You do it over and over and over until you find one that works, but most of the time you're going to follow a protocol that someone else has given you. Protein attachment to the membrane. Also, peripheral proteins can be attached by single or double hydrophobic lipids. Now, if you've got one tiny lipid sitting in there that is attached, but it's not attached very strongly, peripheral proteins tend to be able to be knocked off the outside of membranes pretty easily, and we, by shearing forces, could probably get those proteins off. More commonly, what we do find is that they are attached through charge. And so if we change the pH or change the salt concentration, we can dislodge the peripheral protein from the membranes themselves. But we only isolate them to study them. Now, protein attachment. I take this from one book, and they have what? 6 types of proteins. Okay, you can see. Okay, outside, inside. Does the N terminus go outside? Does the C terminus go outside? Okay, this kind of organization is for a textbook. You never see in a journal article, this is a type 2 protein. No, that's not really said. Okay, so what's important about this? There's a lot of different ways to embed a membrane protein. And so what is a characteristic that we see? All right, we see the characteristic of all of these types, an alpha helix. Okay, that alpha helix must have a very hydrophobic nature, and this. The number one size of a lipid is how many carbons long? What's the most often seen? 16. 16 carbons to the middle. 16 carbons to the middle is the normal distance between a membrane. That normal distance represents 20 amino acids that are in a helical nature. And so, this now gives us a clue to look at a protein and to determine how hydrophobic are the residues and do they most likely are found in an alpha helix. We can plot the primary sequence and plot how polar it is or how hydrophobic it is. If we see bands that are 20 amino acids long, exactly 20 amino acids long, that are very hydrophobic and then become polar, they tend to be the alpha helix that are embedded in membranes. What happens if you find a portion of sequence that is, let's say, 14 carbons long? That's in an alpha helix and very hydrophobic. Does that necessarily mean it is in the membrane? Is it long enough to expand a membrane? No. Is there any other place hydrophobic residues like to be inside a protein? Remember how proteins fold. What happens? They have a hydrophobic collapse. If you're 20, you can span a membrane. If you're 15, you can't. Okay, both of those could be just in the inner core, but a 20 is particularly the right size to be in a membrane. If it's 22, probably not in a membrane. If it's 14, probably not in a membrane. If it is 20, there's a high likelihood that it is a membrane-bound protein. Okay, and it's embedded. And we do see repeating patterns. There are 12 helical proteins that have 12 in them. There's a whole group that have 7. So we see patterns that we can match up just looking at the sequence and measuring, is it hydrophobic or is it hydrophilic? This is called a hydrophobicity plot, a plot of if it's hydrophobic or not, and we can do analysis. Now, we have this nice tool. We can identify alpha helix and be in a membrane, and so we knew how to find all membrane-bound proteins until we get to this. Okay, for a long time, it was thought alpha helix were the ones that actually embedded, and then we started finding a whole host of proteins that were in beta sheets. Beta sheets are much, much harder to identify from the primary sequence itself. Okay, this is called a beta barrel. This is now a very common type of membrane-bound protein. And in actuality, the beta barrel works really well. You see how it's all in there, but let's take a look at this one. And let's look at it from the top. If I'm going to control things to go in and out, what is right there? A hole for things to go in and out. So the beta barrel actually creates a wonderful channel. And so there's a whole host of different. Now, do you see differences about the structures here? Yeah. You've got some that are just right across the membrane, and some that have extra stuff. Okay, well, each one is controlling a certain type of reaction or transport, and so the functionality, it may be the fact that it goes through the membrane, or it may be the protein is just associated to the membrane. All the functions here, this is just an anchor point, whereas this one most likely is a transport protein that allows things to go through. So you can get both, just making sure that it's attached to the membrane, or I am actually functioning to go through the membrane. Now, what we do see in the proteins, or in the lipids, is that at one time we thought all those proteins were just kind of floating around and they would be evenly distributed around the membrane. That was our first idea. But what we did was we started to see something different. Okay? We started to see proteins clump up together, and quite frankly, those proteins all work together in one pathway. And so what we see are called either microdomains or lipid rafts that have proteins that are in one pathway or do one thing organized together. Now, what clusters them together is a lot of cholesterol and certain lipids that are in the membrane, and they organize themselves. So you see a lot of sphingolipid and cholesterol. And so we can even take a X-ray picture of the membranes, and we can see that these rafts or microdomains not only are grouped together, but they're a little bit raised. But really, it is organization as a function of regulation. We group all the things together to make it work better, make it work faster, easier to control. Okay? Microdomains are rafts, and those are pretty specific to create a unique function. Now, of those lipids, we've just been talking about them. Let's take a look at what happens. Most of the time, we kind of cartoonish do this. We say there is some polar group and two fats. That's as detailed as a lot of books go into the lipids, but we're in biochemistry, so we're going to look at the chemistry, what's actually happening. And we talk about all of the polars line up and all the hydrophobics line up, and then we have it on this side and have it on this side, and we have a bilayer because we have two fat groups. Okay? Well, to attach the fats, basically what we have to have is some sort of thing that we attach fats to, and we put two of them on, and then we have to attach something that is charged. Okay, so what we're going to do is look at the two molecules that are kind of the branch point where we put everything on. The first one is glycerol, which is a trialcohol, and it has 1, 2, 3 carbons and 1, 2, 3 hydroxides. Okay, each one of those hydroxides we're going to add fats to, and then we're going to add our polar molecule to make a polar head group. The sphingosine is interesting in that it has attachment points for one fat, and it has one fat incorporated into its own structure. Okay, so I'm going to put another fat on this and then attach a polar head group to make the polar and the two. Now those two molecules don't look really similar written this way, but when you add the two fat groups to glycerol and you add the one fat group to sphingosine, three-dimensionally they almost look identical, but it doesn't look like it in these drawings. And so what these class of lipids are called phospholipids, which are almost the majority of the lipids in membranes. Why in the world do we call them phospholipids? Because we add a phosphate. Okay. Now the phosphate by itself would make that thing polar. Okay. And we will have glycerol and two fats. Okay. Although this is not polar enough, okay, and it doesn't give us enough variation, so to the phosphate we're going to add a whole bunch of alcohols to make them more polar or to give more function. And so of the phospholipids, you can have a class that are called glycerol phospholipids. These are those because I'm using glycerol to attach everything. Okay. The fats are usually numbered 14 to 24, most often 16. They are unbranched. I can have saturated or unsaturated. And the biological diversity is not well understood, even to the point that we don't even classify most phospholipids. We don't tell you what the fats are. What we focus in on is what the polar head group is. And so as long as they're a long chain fat, we really aren't sure why they're there or what diversity gives the membrane. So what you do need to know, though, is the alcohols. These are the most often used alcohols: serine, ethanolamine, choline, glycerol, and inositol. Okay. You've seen choline before, but attached to something, an acetyl group. Where did you see acetylcholine? Wait a minute. That's familiar. It's somewhere in the back of my mind. It's in where? It's in the textbook. Good, that's right, it's in the textbook. Do you know where the acetylcholine is found? It's not part of a membrane. The acetylcholine actually works on a receptor in the nerve junction. The acetylcholine is a neurotransmitter, and it sends a signal from one cell to the next in a nerve cell impulse. Although this choline is just an organic molecule, now will be placed into membrane lipids. Okay? The nomenclature that we have is, okay, we have glycerol, which is the alcohol with three hydroxyl groups. So glycerol. All right, if you get, stop it, if you get, okay, this, and you add a fatty acid, okay, and you get this long thing, okay, you get an O and a double bond and a long fat, and then I'm going to put the hydroxide up here because it's just easier to get it out of my way, okay. This is called DAG, which you may have seen before, diacyl glycerol. Okay, so DAG is diacylglycerol. You've actually seen that in a signal transduction pathway before. If by chance you have a DAG, okay, and you put on a phosphate on there, it's called phosphatidate or phosphatidyl. And then when you put on your polar head group, like this one, phosphatidyl serine, phosphatidylcholine, phosphatidylainositol, phosphatidyl ethanolamine. And so glycerol can be converted into diacylglycerol, DAG, which then can be converted into phosphatidyl, which has a phosphate on the polar hydroxide, or one that will become polar, and then you name the group. All right, good test question. I write this and I put all of those as A, B, C, D. Okay, so being able to recognize them from the structure is about the level that I want you to be able to look at. I will not make you draw them. What is the functional group I just circled in red? An ester. Okay. To create an ester, you learned a organic reaction. If you take an acid plus a alcohol, you will make an ester. Okay. What is the leaving group of that reaction? What do you lose to make the ester? Water. So to break it down, add what? We add water to break down fats. Okay. All right, there is one phospholipid that is special, okay, and it's called an ether lipid. Why? One of the fats is connected by an ether linkage rather than an ester linkage. Actually, chemically, an ether linkage is more stable because it takes more than just water to break that off. Okay, the ester can be broken off fairly easily with water. Now, there are two biggest representatives of this class. One is platelet activating factor. It is not part of a membrane, but it is actually part of a signaling pathway in clotting. If you are absent this, it is one of the many different ways you can have hemophilia. Okay, so this is one ether lipid that we know of. The other ether lipid that is actually in membranes is called plasminogen. It is found almost exclusively in one tissue, and it makes up about 50% of all the lipids in the heart. Now, the heart is working a lot, so maybe that an extra integrity is essential for the muscle of the heart. But we know it's there. We're not exactly sure why. The other class of phospholipids is based upon sphingosine. Sphingosine have classes that are a little bit different. Okay, and so here's the sphingosine. It's got one long hydrophobic tail that's attached first, and then it's attached to this polar group. Okay, sphingosine is actually the condensation of a fat and serine. We take the amino acid serine, add a fat to it, and we get, after a couple reactions, we get to sphingosine. We do add a secondary fat to the amine, and we get two long chains, just like the glycerol. Okay? This hydroxide then can be made a derivative. Now, the two most often alcohols that are added are choline and ethanolamine. They're a lot less diverse in the number of polar head groups that are added. And then there are a whole bunch of them that have sugars attached. Okay? And so we'll look at that. They become a whole different class if they have just sugars attached to them. Okay? We actually get to what are called glycolipids. We're here. So the sugars attached to lipids usually are through the sphingosine. You can attach them through glycerol, but the majority are the sphingosine type that you are probably familiar with. Okay, you're going to hear some words, and I want you to think, what tissue type are you associating when you hear these words? Okay, word, cerebrocyte, ganglioside. Where do you hear those kinds of things? Associated with what? Brain and nerves. These are membrane lipids that are part of the nervous system and in high proportion within the nervous system. Now, what are these? That's great. A cerebrocyte is a sphingolipid with a single sugar. Now, it could be any sugar, but it's just a single sugar. A globuloside is a sphingosine with multiple sugars. A ganglioside is a sphingosine that has multiple sugars but is also charged. So these are the three classifications. And so if you just put glucose on one where the X is up there, it's a specific glucosal cerebrocyte. Okay? If you put a di or tri or just a few sugars on there, it becomes a globuloside. And then if you have a branched sugar or multiple sugars that have charges, it's more considered complex. It is a ganglioside, and then they give them a designate. This is GM2. We're going to have all sorts of letters and numbers that will designate different ones. Okay, and so knowing the three different classifications, how it goes from a definition of a cerebrocyte to a globuloside to a ganglioside is what I would want you to know. I'm not going to throw one out there and ask you for the name of it. Okay. In actuality, the blood typing sugars are glycosphingolipids. And so we talked about A, B, and O. They actually fall into this particular class. All right. If you add a fatty acid and an alcohol, you get that ester, and we can do synthesis. But we also can break them down. And so we have what are called phospholipases. Okay, it works on lipids that have phosphates. Okay, and there are 4 different versions to break down the things that are attached to glycerol. Now, the one thing that you think about is I could break at the first alcohol and the second alcohol and the third alcohol. Okay, that's 3 of them. But there's also one special one in which I can break where I break off the polar head group but leave the phosphate attached. And so if I break out one fat, phospholipase A, it should be phospholipase B, but it's not. It's phospholipase A2. So at the first carbon or the second carbon, phospholipase C breaks off the phosphate and the polar head group, and phospholipase D breaks off the phosphate and the polar head group, leaving the phosphate with the lipid. We have looked at one of these, phospholipase C, in signal transduction pathways, okay, to upregulate, okay, an enzyme called, what is that? Protein kinase version C, okay. Do you remember how to upregulate protein kinase C in the signal transduction pathway? Membrane. Receptor. Do you remember anything about this pathway? No. Okay. A hormone binds to the receptor. Okay. It will interact with a protein that has 1, 2, 3 parts. 3 parts are the alpha, the beta, and the gamma. And the alpha subunit has sitting inside of it a GDP. And when the hormone binds to this, the alpha subunit breaks apart, this is the alpha, and it picks up a what? Does anybody know? GTP. And it's the stimulatory. Do you know what that protein that has the alpha, beta, and the gamma that had a GDP but picks up a GTP, do you know what that's called? A G protein, yes. The G protein will again go and hit another enzyme. Okay? Now we've hit 2 main enzymes. One that's called adenylate cyclase, but the other one that we're going to hit is called PLC, phospholipase C. It will break a specific membrane lipid. It will break a membrane lipid. Okay, the lipid is the target. Okay, and it has a phosphate and a inositol. Okay, that has 2 phosphates on it. And the PKC breaks it on this side of the phosphate. Break it into 2 things. I break it into something called IP3, and I break it into something called DAG. Okay? DAG is part of the membrane. It goes to an enzyme called PKC, okay, and it stimulates it. Do you know where IP3 goes? Anybody remember? I'm going the Wayback Time Machine. Goes to the ER. What's inside the ER? Calcium, not G. what in the world's going on? Calcium 2 plus. And it releases the calcium 2 plus, not G. The calcium, ooh, it really went back. Ah, it's releasing the calcium 2 plus, which interacts with the PKC. And when the DAG binds to the PKC and the calcium, the PKC becomes active and phosphorylates things, and we get what's called a cellular response. Okay? But in all of that, what we taught you here is this enzyme right here, the PKC. The PLC. It's a phospholipase C. It actually breaks that. Now, we learned that pathway. We also learned how to upregulate pKa. Okay, those are probably the two most often taught signal transduction pathways. And if they're the most often taught in every textbook, they're probably the most often tested ones on standardized tests. Okay. Now, lipid membranes have all of those. It has sphingosine base and have the phospholipids, that's the glycerol base, but we also have the steroids. And steroids are part of the membrane. But steroids are not only functioning as part of the membrane. Okay? When I put steroids in a membrane, when I put cholesterol there, what do I do to the fluidity of a membrane? Do I increase it? It can go either way. It's a modulator, keeps it in the middle. If I add saturated, I make it more rigid. If I add unsaturated, it's more fluid. Okay? But there's a whole host of functions that cholesterol also does. Okay? Not just be embedded in the membrane. If you take a look at the cholesterol, it is really kind of strangely numbered. It actually makes sense for a little while, then it goes crazy. 1, 2, 3, 4, 5, that seems good. 6, 7, 8, 9. And then all of a sudden, it goes 10. Okay, that seems right. And then it has nowhere to go, so it goes 11, 12, 13, 14, 15, 16, 17, right? And then, obviously, it goes 18, and then 19, and then 20, and, 21, and then up or down. Okay, do you have to know all of that? No. Why? Because you can always look it up. Except when you're given a graduate level CUME final, which I should have known the numbering, and I didn't do well on that test. But I didn't know the person who was writing the test studied cholesterol as his main thing. So you need to find out if you're ever doing graduate work. There are times in which you do a written analysis. Are you making progress? You find out who's going to write the CUME that month. You look at their research, because it's probably going to be part of their research something, and see what kind of class they are teaching. And look at not what you're studying in class right now, but look at a week ahead, because that's where they're prepping. Okay, and they're going to take something out of what they're prepping to teach and their research, and that's what they're going to put on, because they can take anything in chemistry, whatever you want. All right. Cholesterol can be made derivatives of. Okay. And the derivatives have certain functions. The first thing that happens to a cholesterol in terms of it becoming functionalized, it gets processed by the liver, and you start to add hydroxides all over it. If you're trying to get rid of cholesterol, what you have to do is make it soluble. Okay? And to make it soluble, you add hydroxides. And they're going to add on a hydrophobic entity at the end. So what you can see is polar and polar and polar and polar, and what's in the middle? Nonpolar. The type of molecule that you get here is called bile. Okay? And bile is used to solubilize or emulsify fats as you eat them. Okay? And That is great. If you eat a really fatty meal, you have an organ that dumps bile into your stomach. What is that organ? The gallbladder. Okay. If you have one. Okay. I tell you, I was walking at work and I felt like I was having a heart attack. It felt so bad. I went to the doctor. They put an EKG on and everything, and like, nope, your heart's fine, but your gallbladder is not. Okay, and you know when you go to the techs to do analysis, so I went to the sonogram tech, and he does gallbladders all the time, and they can't tell you anything, okay, because the doctor has to tell you something. Although the person's done it for 20 years, they know what they're seeing. They could actually tell you what's going on. He turned over to me, and obviously he saw some stones. And he looked at me and he says, "How much pain are you in?" That didn't make me feel good, okay? I guess my stones were very, very large. The best feeling I have ever had, about the second best feeling I've ever had, was the removal of my gallbladder, because it was so much better. The worst was, here's another story. We used to have an internship where all health science majors actually had to work at what is now Tenova. And so it was called something way back when. And a lot of times they worked in the emergency room. And so I am at home, again, all of a sudden, pain. Now I knew what this pain was. The only remedy to this pain is morphine, and that's a kidney stone. So I have my wife drive me to the hospital. She hits every hole in the road from our house to the hospital. And I'm in a fetal position on the gurney waiting for the doctor to come see me. I hear the nurse, 2 students to the side. Now, we're going to cover some HIPAA laws. Just because he is your professor doesn't mean that you can tell everybody that he's here. After I got the morphine, I didn't care anymore, and so that was okay. So that isn't as bad. I'm going to tell you one more story. I'm going to have to cut this totally out. I know of a professor who, in a small town, maybe going for a procedure might not be always great. So he walks in to the doctor to have a procedure. He and his wife decided not to have any more children, so you can imagine what procedure it was. And the receptionist says, "Oh, hi, Dr. So-and-so." Not what you want to hear going in for that kind of procedure. Snip, snip. All right. So, privacy. Okay. So bile salts are actually part of digestion and the degradation of cholesterol, but as part of that process, you can also have the hydrophobic tail cleaved off, and we make derivatives into what are called the steroid hormones, which are very powerful regulators. One of the first hormones that we actually identified. They're actually very large in concentration. Hormones that were some of the first drugs used medically, still used today, but they act globally. The conversion of hormones all go through pregnenolone, which is the common hormone precursor, and then every type of cholesterol-based hormone or steroid-based hormone, including the testosterone and the testosterone and the estrogen are part of these. And so, but all of the steroid hormones are smaller in size than cholesterol itself. There are a whole bunch of classes that have a whole bunch of physiological responses. And so, but all based upon cholesterol. So cholesterol is not necessarily bad. Cholesterol is actually necessary. And so we will use cholesterol in a number of different ways. Okay. There are also a lot of vitamins that are based upon derivatives of fats. And one of those is cholesterol itself and vitamin D. Oh, do I hear something? I do not hear anything. Okay, vitamin D actually has a couple things before it actually becomes active. There is the kidneys and the liver that are part of the process. And what we have is the cholesterol derivative, before it becomes vitamin D, actually has to be worked upon by sunlight to actually break a ring. This broken ring structure is still not active. We have to have a hydroxide placed in two positions, here on the chain and here on the ring, and that only occurs as the cholesterol goes through both the kidney and the liver. Once you have the liver, we have the hydroxide placed here. Once you go through the kidney, we have the hydroxide placed here. And so the sun and the processing between two different tissue types actually creates the active calcium-controlling vitamin, which is the vitamin D3. Now, vitamin D3, or vitamins like the vitamin D, that is actually one that controls calcium regulation, is actually becoming a vitamin that is in deficiency in most of the population in the U.S. Why do you think that is? We don't go outside anymore. We don't get enough sun. Even though we supplement it in the milk, the vitamin D has to be placed in milk. It gets in there, but it never gets processed into the right way to be the active form of vitamin D. Other lipids that function, okay, are messengers or cofactors, and we'll look at some of those. Here's that pathway for the phospholipase C activation. What we actually have is phosphatidylinositol that gets phosphorylated a couple times, and then when it breaks apart by phospholipase C upon the hormone, you get this inositol with 1,4,5-triphosphate. We call that IP3, and we call diacylglycerol DAG. Okay, so IP3 and DAG are these particular molecules. And so they can be, the lipids themselves can be signaling molecules. Okay, there's a whole host of messengers that are derived from fats called the eicosanoids. Now, eicosanoids are not actually hormones. I use the term hormone because you know what that means. A hormone is actually a small molecule that is released in the bloodstream that affects the entire body system. An eicosanoid is a small molecule that is given out and it only affects very short neighbor-to-neighbor cells. Okay? So it doesn't go through the entire body and the blood system. And so these particular fats are derived from arachidonic acid, which is 20 amino acids long with 4 unsaturations. Arachidonic acid is actually an essential fat because the unsaturations in the 11th and 14th position cannot be done by humans. So this has to be done in plants, and we have to eat this particular fat to live. Now, how many people think of plants as being fat? Yeah, if you eat avocado, there's a fatty plant. Okay, so there are some plants that have fats derived within them, but they aren't necessarily the fats that we think about. Okay, we're going to have 4 classes of eicosanoids. We will pick up at the next time that we come together at the end of this chapter and then start the next chapter